In mammals, the egg is small and yolkless. It develops inside the body of mother and obtains its nourishment from uterine wall. For this purpose, the embryo develops connection with the maternal uterine wall, which is known as placenta. Depending upon the mode of origin, structure and distribution of villi, various kinds of placenta have been differentiated in mammals. These are: first is non-deciduate, second is deciduate, and the last is contradeciduate. Non-deciduate type. In this type of placenta, the contact between the foetal and uterine wall is not very intimate. The villi are simple and unbranched, and they lie in the crypts of the uterine wall. Thus, it is also known as semi-deciduate placenta. At the time of birth, the villi are completely withdrawn, causing no injury to the uterine wall. According to the manner of distribution of villi, the non-deciduate placenta may be of two types. First is diffused and the second is cotyledonary type. Diffuse type. The villi are diffusely scattered all over the chorion, for example horse, donkey, etc. In cotyledonary type, the villi are restricted to the patches or cotyledons, for example in goats or all ruminant animals. Deciduate. In deciduate placenta, the villi are branched and deeply embedded in the crypts. These are cause damage to the uterine wall. At the birth, when the villi are withdrawn, the vascular part of the maternal tissue also gets ruptured and comes out accompanied by bleeding. Depending upon the distribution of villi, the deciduate placenta may be of three types: Joindre, desicquoidal, metadesicquoidal. Joindre. The villi are arranged in definite band or girdle or zone around the embryo. It is found in carnivorous mammals such as cats, dogs, tigers, lions, etc. Discoidal type. The villi form a disc-like patch on the dorsal surface of the embryo. It is found in rodents like rabbits, rats, moles, etc. Metadiscoidal type. The villi are diffused at the first but becomes restricted to the small disc-like area on the ventral side of the embryo. It is found in human, apes, etc. Contradeciduate. In this type of placenta, connection between the foetal villi and uterine crypts becomes too intimate that at the time of birth a portion of allotic placenta is left behind and is absorbed in the uterine wall. That is found in bandicots and moles.
Development of the placenta begins during implantation. When the blastocyst adheres to the endometrial epithelium, trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytiotrophoblast layer. As the cytotrophoblast proliferates, newly formed cells migrate into the syncytiotrophoblast and lose their cell membranes, forming a rapidly growing multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytiotrophoblast sends out finger-like projections, allowing the blastocyst to embed into the endometrium. Lacunae, or spaces, begin to form within the syncytiotrophoblast. As the syncytiotrophoblast erodes the endometrial blood vessels and glands, the lacunae become filled with maternal blood and glandular secretions. Isolated lacunae fuse to form a network through which maternal blood flows, thus establishing early uteroplacental circulation. Toward the end of the second week, small projections of the cytotrophoblast begin to expand into the syncytiotrophoblast, forming primary chorionic villi. Early in the third week, extraembryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point the structures are known as secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the extraembryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. Cytotrophoblast cells from the tertiary villi grow toward the decidua basalis and spread across it to form a cytotrophoblastic shell. Villi that are connected to the decidua basalis through the cytotrophoblastic shell are called anchoring villi. Villi growing from the sides of the stem villi are called branch villi. These branch villi are surrounded by the intervillous space and will serve as the main sites of exchange between mother and fetus. By the 4th week, fetal blood flow is established. Poorly oxygenated blood is transported to the placenta from the fetus via paired umbilical arteries. These arteries send branches into the chorionic villi and divide into capillary networks at the terminal ends. Carbon dioxide and waste products are removed from the fetal circulation across the placental membrane to the maternal blood in the intervillous space. Oxygen and nutrients are transported across the placental membrane from the maternal blood to the fetal capillaries. Oxygenated fetal blood travels back through veins, converging to form a single umbilical vein that goes to the fetus. Blood returns to the maternal circulation through endometrial veins, forced out by the pressure of incoming arterial blood. The placenta nourishes the fetus throughout development and is delivered shortly after parturition.